Today we're going to look at diffusion and osmosis. Now, anytime you've smelt dinner cooking in the kitchen, that's diffusion. Anytime you've smelt the aftershave or deodorant of your family members, that's diffusion. Diffusion is the movement of gas or liquid particles from an area of high concentration to an area of low concentration. This means from where there's lots and lots of it to where there isn't very much. In the case of the dinner cooking in the kitchen, there are lots and lots of gas particles around that food in the kitchen. These particles then move, spread out, randomly move to fill the whole room, or in the actual case of you smell upstairs, the whole house. This is diffusion. Those gas particles were all tightly packed and they spread out through chemistry, random motion, to fill the whole area. Likewise, you put on aftershave or perfume, within a few minutes those around you can smell it, because those gas particles spread from your body in high concentration and they move to a lower concentration. They equal out. This is called equilibrium. So diffusion is a movement of gas or liquid particles from an area of high concentration to an area of low concentration. Now, as you know in chemistry, the movement of particles in gas and liquid, this happens faster in a gas than in a liquid. Why is diffusion important in the real world? Well, cast your mind back to year 8, the breathing system. We inhale and exhale air. The composition of this air changes. The air you breathe in is high in oxygen. This diffuses from the alveoli into your blood, and the air you exhale is high in carbon dioxide. This has come from the blood into the alveoli and diffused out. Also think about digestion. The digested food products are all absorbed across the villi and microvilli via diffusion. And finally, plant cells. Plants carry out photosynthesis and respiration, in which case carbon dioxide and oxygen are either entering or exiting the plant down their concentration gradient via diffusion. Now, I think we've established diffusion is pretty awesome. There is a special type of diffusion which happens within water. This is called osmosis. Now, if you imagine that there is a very salty solution of water full of salt, and this is separated from a pure water source by a thin membrane, a partially permeable membrane. Hopefully it stands to reason that those two liquids will mix and dilute each other until they are both equal concentration. This is osmosis. Just like diffusion, moving particles from an area of high concentration to an area of low concentration, water, providing there is a membrane between them, will follow the same rules. Water will move from an area of high concentration, the purest water, to an area of low concentration, the most concentrated or least dilute water solution. This is osmosis. This can happen in all living cells, providing there is a selectively permeable membrane. So, we know from year 7 that plants have a cell membrane and a cell wall. We know that that cell wall is super strong. Let's imagine then that I place a plant cell into a very strong salty solution, extremely strong. This will cause the water from the plant cell, from the vacuole, to leave that plant cell and enter the surrounding solution. This is termed a hypertonic solution, very salty, very strong. The water has left the plant cell and has entered the solution around it. This has made the cell plasmalized. It has lost water. The vacuole and the cell contents have shrunk. The cell is plasmalized. Let's imagine the opposite scenario. I place a plant cell into a pure water solution. Now, a plant cell has lots of water, lots of minerals, but it is not pure water. This would mean that water from the surrounding solution would enter the plant cell and fill up the vacuole. This would mean the cell squashed up like a big water balloon, but it doesn't burst. Why? There is a plant cell wall. So our cell becomes turgid. This means that water has rushed in, filled the vacuole via osmosis, but the plant cell does not burst because it is turgid. If we get the best scenario, an equilibrium, this is called isotonic solution, thereby water enters and exits our plant cell in a basically equilibrium balanced medium. We call the cell flaccid. This means water is moving in and is moving out, but it is about equilibrium. The same in and the same out. The cell is flaccid. Now we'll look at the same scenario, but with animal cells. Now immediately your brain should start whirring through the idea that animal cells don't have a cell wall. So the outcome will be slightly different. Let's imagine red blood cells. If we take a red blood cell and we place it into a hypertonic, very salty or very strong solution, this will cause the cell to lose water. The cell will shrivel up, the cell membrane will collapse down, and the cell will become what we call hemolyzed. Now, red blood cells are called hemolyzed when they lose water. Any other animal cell would be termed crenated. Crenated is the procedure whereby a cell loses lots of water and shrinks up. The special name for it in a red blood cell is hemolyzed. If that red blood cell or animal cell is placed into an isotonic, equilibrium-based solution, water will enter the cell, water will leave the cell in equilibrium, and the cell will maintain a normal shape. If that cell is placed into a hypotonic, very wet or very watery solution, water will enter the red blood cell. As a result, the cell will swell. Now, it has no cell wall, so as it heads towards becoming turgid, it ruptures. There is no cell wall pushing back against the red blood cell or any animal cell, and therefore, when water enters from a hypotonic solution, the cell will swell and then burst. This is osmosis in animal cells. Big difference to plant cells because there is no cell wall.